Welcome to Learn While Sleeping. Let your jaw soften, let your shoulders drop, and breathe in and out. You don't have to try. You don't have to remember. Just rest and let the rest of the story drift by like simple pages turning. Let all the details wait. All you need is the shape of the journey, from wondering what each thing is to asking what qualities make each thing what it is, to following evidence carefully until the answer comes into focus. Feel your breath slow. Imagine a softly lit lab bench, glassware resting, notes closed, while the ideas remain simple, steady, unhurried. You are safe to drift. There's no test, no exam. When you wake, everything and the story will still be here. Don't worry. Tonight, we drift into the quiet but constant world of the body's innate defenses. Imagine a system that has no choice but to be ready, always, waiting for the moment when a microbe slips past the surface barriers of skin and mucosa. This is the induced innate immune response, the body's second layer of protection, mobilized when an invader has managed to bypass the fixed immediate shields. The first actors in this response are cells you've carried your entire life: macrophages, neutrophils, natural killer cells, and dendritic cells. They don't wait for training. They don't rely on memory. Instead, they hold onto tools crafted through evolution, tools designed to recognize the common shapes and textures of microbes. These shapes are called pathogen-associated molecular patterns, or PAMPs. They might be the repeating sugars on a bacterial wall, the lipopolysaccharide that gives gram-negative bacteria their distinctive armor, or viral nucleic acids that betray infection. To recognize these microbial signatures, immune cells carry pattern recognition receptors, or PRRs. Unlike the intricate antigen receptors of adaptive immunity, PRRs are simple and broad. One receptor can recognize an entire category of molecules shared across many microbes. It is as if a security guard were trained to look for any red passport rather than memorizing the face of every possible visitor. Macrophages, in particular, are loaded with such receptors. Scavenger receptors cling to lipoteichoic acids on bacterial walls. Mannose receptors, part of the lectin family, reach out for carbohydrate chains that are common in microbes but rare in human tissues. Each receptor does more than simply bind. It initiates a response, tugging the microbe inward, folding the plasma membrane around it, and delivering it into an acidic vesicle. This vesicle is the phagosome. Within the cell, the phagosome merges with the lysosome, creating the phagolysosome, a contained storm of acidity, enzymes, antimicrobial peptides, and reactive molecules. Oxygen is consumed rapidly, fueling bursts of superoxide and nitric oxide radicals, corrosive weapons aimed at dismantling the invader piece by piece. The microbe is broken down until only fragments remain. But the process is more than destruction. Each engulfed particle becomes information. Dendritic cells in particular use these captured fragments to signal onward, preparing the adaptive immune system to engage with precision. Even in this early moment, the innate immune system is not only clearing infection, but also shaping the battle that lies ahead. As you breathe slowly, imagine those first sentries at work, macrophages, listening with their pattern recognition receptors. Neutrophils, waiting to sprint into tissue. Dendritic cells, preparing messages for the adaptive system. A basic, vigilant watch, always ready, always steady. Part 2. Toll-like receptors, turning danger into signal. As microbes slip into tissues, recognition has to go beyond just swallowing them. Some receptors are made to signal, to set in motion cascades of instructions that ripple through the immune system. These are the toll-like receptors, or TLRs. Think of a TLR as a doorbell on the surface of the immune system of an immune cell. Each one is tuned to ring in response to a very specific microbial feature. TLR4, for example, recognizes lipopolysaccharide, the outer wall component of gram-negative bacteria. Structurally, TLRs have a curved extracellular domain made of leucine-rich repeats. This region is like a horseshoe designed to cradle its microbial ligand. Inside the cell, the TLR has a TIR domain, or toll interleukin-1 receptor domain, which is built for passing on the signal. Here's how it plays out. A macrophage meets a bacterium coated in LPS. LPS first binds to LPS binding protein in the fluid. Then it hands it off to CD14 and MD2, which helps present it to TLR4. Once TLR4 is engaged, the inside of the receptor changes shape. That movement recruits adapter proteins and kinases, like little signal carriers. One of the key outcomes is the activation of the IKK complex, the IKB kinase. IKK's role is to deal with a protein called IKB. Normally, IKB keeps the transcription factor NFKB trapped in the cytoplasm, like a guard keeping someone locked in a waiting room. But when IKK is switched on, it phosphorylates IKB, tagging it for destruction. NFKB is freed. Now, NFKB can travel into the nucleus of the cell, into the library where genes are stored, and bind to specific DNA sites. There, it turns on genes that encode inflammatory cytokines. Cytokines like TNF, IL-1, and IL-6, which all act as distress signals, spreading the word to neighboring cells, making blood vessels more permeable, and recruiting neutrophils to the scene. This cascade is powerful, but it must be tuned carefully. NFKB isn't just an on switch. It's context dependent. Context dependent meaning that it responds differently depending on what cell type it's in and what other signals are present. For example, if there's too much activation or faulty regulation, well then it can lead to disease. For example, mutations in the gene encoding NEMO, a critical subunit of the IKK complex, prevent NFKB from being activated. People with NEMO deficiency suffer from recurrent severe infections and problems with the development of skin, hair, and teeth. On the other hand, overactivation of NFKB is linked to chronic inflammation and cancer. So, the body balances, allowing enough response to clear infection, but not so much that it harms itself. Just try to picture it this way. Toll-like receptors are like neighborhood alarms. They don't fight the fire themselves. Instead, they trigger the release of signals, they call for backup, and they coordinate the arrival of the immune system's next wave. Now as you rest, you can imagine those signals quietly pulsing out from a single cell, NFKB stepping out into the nucleus, turning on the lights of cytokine genes, and preparing the neighborhood for defense. Part 3. A family of toll-like receptors. Toll-like receptors are not a single sensor, but actually a family of sensors. Each one has its own specialization, like different smoke detectors tuned for heat, carbon monoxide, or gas. TLR4, as we said, is tuned to lipopolysaccharide from gram-negative bacteria, but other TLRs expand the surveillance system. Some are positioned on the cell surface, ready to detect bacterial and fungal products. Others are embedded in endosomal membranes, where they sense viral nucleic acids once a pathogen has been swallowed. For example, TLR2 recognizes lipoteichoic acids and other wall components of gram-positive bacteria. TLR3 detects double-stranded RNA, a common intermediate of viral replication. TLR7 and TLR8 sense single-stranded viral RNA. TLR9 is specialized for unmethylated CpG DNA motifs, which are common in bacterial and viral genomes, but rare in our own DNA. Each of these receptors share a similar architecture: the extracellular leucine-rich repeat region for recognition, and the intracellular TIR domain for signaling. But what differs is the ligand that they bind and the adapters they recruit inside the cell. Some pathways activate NF-κB, some activate IRF transcription factors to induce interferons, and many do both. An important detail to remember is that variability exists among TLRs in the human population. Genetic polymorphisms are more common in TLRs that recognize surface determinants, like TLR2 and TLR4, compared to those that sense nucleic acids. That makes sense because nucleic acid recognition must be very precise. Too much variation risks mistaking our own DNA or RNA for viral patterns. Clinically, this variability can matter. Some polymorphisms in TLR4, for example, alter sensitivity to bacterial LPS, which can change how strongly a person's immune system responds to gram-negative infections. This actually shows how innate immunity is not identical from one person to the next. Together, the TLR family builds a layered detection network. They don't work in isolation. They connect with phagocytic receptors, with nod-like receptors in the cytosol, and with interferon pathways. They are the crossroad sensors, triggering inflammatory cytokines, chemokines, and antiviral defenses in a coordinated way. As you drift off, imagine this invisible array of sensors spread across your tissues, some standing guard at the surface, others waiting inside endosomes. Each one is quietly tuned to recognize a different molecular whisper of infection, ready to send the signal that awakens the rest of the immune system. Part 4. Weaving the induced innate response. Now that we've walked through the details, let's step back and see the whole pattern of the induced innate response. It begins when a microbe breaches the immediate barriers, the skin, the mucosal lining, the chemical shields of secretions. At this point, the early fixed defenses have failed. And now the induced response must act. First, immune cells like macrophages and dendritic cells use their pattern recognition receptors, or PRRs, to detect microbial signatures. These are PAMPs, lipopolysaccharides, peptidoglycans, mannose-rich sugars, or viral nucleic acids. Receptors like scavenger receptors and mannose receptors both recognize and internalize these invaders through phagocytosis. Inside the cell, phagosomes fuse with lysosomes to form phagolysosomes, where acidity, enzymes, antimicrobial peptides, and radical oxygen species dismantle the microbe. Second, recognition by signaling receptors, especially toll-like receptors, sends messages inward. Engagement of TLR4 with LPS, for example, recruits adapter proteins, activates the IKK complex, and frees NF-κB to enter the nucleus. Once inside, NF-κB turns on the genes for inflammatory cytokines like TNF, IL-1, and IL-6. These cytokines spread outwards, telling blood vessels to dilate, making tissues more permeable, and summoning neutrophils to the scene. Third, this signaling is tuned and regulated. NF-κB is not a simple switch. It's actually context dependent, responding differently depending on cell type and circumstance. Without regulation, the system risks either underreaction, as seen in immunodeficiency, where NF-κB can't be activated, leading to severe infections and developmental problems, or overreaction, where too much of the NF-κB drives chronic inflammation or cancer. Finally, the family of TLRs provide coverage across different threats. Some watch the surface for bacterial components, others peer into endosomes for viral nucleic acids. Their variability across the human population introduces both strengths and vulnerabilities, shaping how different people respond to infections. The induced innate immune response, then, it's not brute force. It is a carefully layered system. Recognition by PRRs initiates phagocytosis. Signaling by TLRs triggers NF-κB and cytokine production. Together, these processes form an inflammatory response that contains infection and prepares the ground for adaptive immunity. As you breathe now, picture this flow like concentric ripples in a pond. Recognition at the center, signaling and cytokine releasing, spreading outwards, and finally, the broader immune system being drawn into action. A system built on vigilance, tuned for balance, and designed to keep you safe. Part 5. The secret dance of innate immunity. The induced innate immune response is like a secret dance between recognition and reaction. A macrophage meets a bacterium and senses its foreign sugars. A mannose receptor guides it forward, pulling it into a vesicle where enzymes and radicals quietly dismantle the intruder. At the same time, toll-like receptors pick up the alarm, turning recognition into signal. Inside the cell, a chain of kinases awaken, releasing NF-κB to step into the nucleus and turn on the genes for inflammation. Cytokines begin to spread like ripples, soft messages carried to neighboring cells and through blood vessels. They loosen the vascular walls, summon neutrophils, and prepare tissues for defense. Dendritic cells collect fragments of invaders, carrying them away to present to the adaptive immune system, which waits like a more specialized army in reserve. Even here, balance is everything. Too little signaling, and infections persist. Too much, and the body risks burning itself with its own flame. Mutations, like those affecting Nemo, show us what happens when this balance is lost. But most of the time, the system works as designed, fast, reliable, and tuned through million years of evolution. As you rest now, you can imagine this system working in silence inside you, day and night. Receptors that never sleep, cytokines that whisper between cells, an ancient choreography, unseen but constant, keeping the body's inner world safe and steady. Part 6. Interferons, the body's viral alarm. Now we turn to the challenge of viruses. Unlike bacteria, which live outside cells and can be swallowed whole, viruses hide inside our own cells, using our own machinery to replicate. The body needs a different kind of early response here, one that warns neighboring cells before the infection can spread. That response is the interferon system. Inside the cytosol, immune sentinels called RIG-I-like receptors, or RLRs, constantly scan RNA. They look for subtle signs of non-self, a missing cap, a triphosphate group at the 5' end, or double-stranded RNA that rarely appears in healthy human cells. When RIG-I binds such viral RNA, it changes shape and signals through an adapter called MAVS, or MAVS, found on the mitochondria. This signal awakens two transcription factors, IRF3 and IRF7. Together, they switch on the genes for type 1 interferons, specifically IFNA and IFNB. This means IFN alpha and IFN beta. These interferons are barely detectable in healthy people, but during infection they surge, spreading through tissues as antiviral messengers. Now, what do interferons do? They bind to receptors on almost every nucleated cell. Those receptors are always present, waiting, so the response can be immediate. When activated, they turn on hundreds of interferon-stimulated genes. These genes make the cell a poor host for viruses. They degrade viral RNA, block viral protein synthesis, and stiffen the membrane against budding virions. They also increase antigen presentation, ensuring that the adaptive immune system will be alerted more quickly. There are almost all kinds of different families of interferons. Type 1 interferons, IFN alpha and IFN beta, are made by almost all infected cells. Type 2 interferons, we have IFN gamma, which is different. It is produced by T cells and natural killer cells, and its main job is to activate macrophages. Together, they form a layered defense. Some cells are especially powerful interferon producers. Plasmacytoid dendritic cells, sometimes called professional interferon producing cells, can release enormous amounts of type 1 interferons when they detect viral material. These cells circulate in the blood and lymphoid tissues, ready to flood the system with antiviral signals. This interferon storm may sound chaotic, but it is highly coordinated. Each interferon molecule carries a message from one cell to the next. A virus is here. Prepare yourself. It is the body's way of buying time, slowing the infection, so that the adaptive immune system can rise to full strength. As you breathe now, imagine this system in action. A single infected cell notices viral RNA. Within minutes, it releases interferons, and nearby cells become resistant, shutting their doors to viral replication. It is a neighborhood pulling together, not in panic, but in absolute preparation. Part 7. The inflammasome and fiery death. Viruses aren't the only challenge. Some dangers live inside cells in other ways. Bacteria hiding after phagocytosis. Toxins, even crystals or stress signals. To sense these hidden threats, the body has another system, the inflammasome. At the heart of this system are nod-like receptors, or NLRs. These are cytosolic sensors with 3 parts: a leucine-rich repeat region for recognizing danger signals, a nucleotide binding domain for self-assembly, and an effector domain for recruiting other proteins. One of the most studied is NLRP3. Activation of the inflammasome requires 2 steps. First comes the priming step. Signals from toll-like receptors, for example, induce the cell to make the inactive forms of NLRP3 and pro-IL-1 beta. These are stored waiting. Then comes the trigger. Things like bacterial wall fragments, ion flux, reactive oxygen species, or even crystals of uric acid. When the trigger appears, NLRP3 proteins cluster together, pulling in adapter proteins and many molecules of procaspase 1. By gathering so many procaspase 1 molecules close together, they auto-activate. Now, active caspase 1 can cleave pro-IL-1 beta into IL-1 beta, the master regulator of inflammation. IL-1 beta is a potent cytokine. It increases vascular permeability, induces fever, and recruits waves of neutrophils. But IL-1 beta is not secreted by the usual pathways. Instead, the inflammasome creates pores in the cell membrane through a protein called gasdermin D. These pores let IL-1 beta out, but they also disrupt the cell, leading to a fiery, inflammatory cell death called pyroptosis. Pyroptosis is very different from apoptosis, the silent and tidy cell death used in development and tissue renewal. Apoptosis packages a cell neatly into vesicles to be eaten quietly. Pyroptosis is noisy, messy, and pro-inflammatory by design. It sacrifices the infected macrophage to alert the tissue, release IL-1 beta, and call in reinforcements. This system is powerful and potentially dangerous. If inflammasome activity is not controlled, it can lead to autoinflammatory diseases. These are conditions where the innate immune system attacks the body without any infection present. Patients suffer recurrent fevers, rashes, joint pain, and systemic inflammation. Unlike autoimmunity, which involves T or B cells mistakenly recognizing self-antigens, autoinflammatory syndromes are driven by innate sensors misfiring. One example is NOMID, neonatal onset multisystem inflammatory disease. In this rare condition, mutations in the NLRP3 gene cause excessive caspase 1 activation and constant IL-1 beta secretion. Babies are then born with recurrent fever, skin rashes, and joint inflammation. Treatment of this disease involves blocking IL-1 receptors with drugs like anakinra, which prevents IL-1 beta from signaling. Even common conditions like gout connect to this pathway. Monosodium urate crystals deposit in joints, activating NLRP3, releasing IL-1 beta, and triggering neutrophil-driven inflammation. The result is the painful swelling of a gout flare. So, The inflammasome is both a hero and a hazard. It helps clear hidden infections with explosive inflammatory signals, but when unregulated, it sets off disease. As you rest now, you can imagine this system like a fire alarm inside a cell. Most of the time, it is silent, but when it detects danger, it sounds, releases signals, and even allows the cell to burn itself out for the greater good. A fiery sacrifice, restoring balance to the tissue. Part 8. Neutrophils, the sprinters of immunity. When an infection takes hold in tissue, one of the earliest responses is the recruitment of neutrophils. These are the most abundant white blood cells in circulation, but under healthy conditions, they remain in the bloodstream. They are like sprinters on the starting line, waiting for the signal to run. The signal comes from chemokines, small messenger molecules released by infected tissues and activated macrophages. Among the most important are CXCL8, also known as IL-8 and CCL2. These chemokines diffuse outwards, creating a gradient. Neutrophils sense the gradient through receptors and begin to move towards the higher concentration, homing in on the infected site. Getting there requires crossing the wall of the blood vessel, a process called extravasation. First, neutrophils roll along the endothelium, loosely binding and releasing through interactions with selectins. Then, integrins on the neutrophil engage tightly with adhesion molecules on the vessel wall. Finally, the neutrophil squeezes between endothelial cells and enters the tissue. Once inside, neutrophils unleash their arsenal. During their development in the bone marrow, they were loaded with 4 types of secretory granules. These granules contain proteases, myeloperoxidase, defensins, and other bactericidal agents. When a neutrophil engulfs a bacterium, the granules fuse with the phagosome, releasing their contents into the enclosed space. Another weapon is the respiratory burst. Using the enzyme NADPH oxidase, neutrophils rapidly consume oxygen and generate reactive oxygen species. This raises the pH of the phagosome and activates enzymes that kill the microbe. This process is intense but short-lived, designed for rapid clearance. Neutrophils can also release NETs, neutrophil extracellular traps. These are webs of decondensed chromatin mixed with antimicrobial proteins. By casting these sticky nets, neutrophils can immobilize bacteria and fungi, preventing them from spreading while other immune cells finish the job. Neutrophils are not built for longevity. After performing their function, they undergo programmed cell death. Their remains are cleared by macrophages, preventing excessive tissue damage. Even if their death is part of the plan, ensuring that inflammation eventually resolves. Clinically, neutrophils are central to the signs of inflammation: redness, heat, swelling, and pain. Increased blood flow brings warmth and color. Vascular permeability allows plasma and cells to enter tissues, causing swelling and pressure on nerves, which creates pain. It is the visible, sometimes uncomfortable evidence of an immune system at work. As you breathe slowly now, imagine these cells like a flood of first responders. They roll out of the blood vessels, chase chemokine trails, and attack invaders with speed and precision. They don't linger. They're designed to burn bright and fast, clearing the path for the slower but more specialized adaptive immune system to arrive. Part 9. Inflammation, Resolution, and Balance. With macrophages sounding alarms, inflammasomes releasing IL-1 beta, and neutrophils sprinting into tissues, the innate immune system sets the stage with a process we recognize as inflammation. The 4 classical signs are easy to see. Redness and heat come from increased blood flow as vessels dilate. Swelling comes from plasma leaking into tissues, an accumulation called edema. Pain comes from that swelling pressing on nerve endings, and from chemical mediators sensitizing nerves. Together, these changes are not mistakes. They are strategies to slow infection, concentrate immune cells, and prepare for repair. Let's go over those 4 classical signs one more time. Redness and heat come from increased blood flow as vessels dilate. Swelling comes from plasma leaking into tissues, which is an accumulation called edema. Pain comes from that same swelling pressing on nerve endings, and from chemical mediators sensitizing nerves. But as with every immune pathway, balance is key. When inflammation is carefully regulated, tissues return to normal, cytokines quiet, vessels reseal, and neutrophil corpses are cleared away. Macrophages shift from producing inflammatory signals to producing growth factors that encourage healing. This transition is how a crisis becomes resolution. When regulation fails, however, inflammation itself becomes disease. In autoinflammatory conditions, the innate immune system attacks without infection. Mutations in inflammasome components like NLRP3 can keep IL-1 beta flowing constantly. Patients experience fevers, rashes, joint pain, and systemic inflammation, sometimes from birth. In gout, for example, crystals activate the same pathway, triggering painful inflammation in the joints. So, the story of the innate immune system is not just one of attack, but actually one of restraint. It's about knowing when to start the fire and when to let it go out. As you settle into rest, picture this balance like a neighborhood returning to calm after an emergency. The alarms are silenced, the responders pack up their tools, and the streets clear. What remains is a memory carried forward to shape future responses, and a restored sense of quiet. Your body practices this balance every day, vigilant, responsive, and ready to reset. A system that protects you without you noticing, so you can drift safely into sleep. Part 10. The Symphony of Innate Immunity. The innate immune system is your body's first organized response when barriers are breached. It does not need training, and it does not wait. Instead, it relies on ancient tools shaped by evolution. It begins with recognition. Cells like macrophages and dendritic cells carry pattern recognition receptors, or PRRs, tuned to sense molecular signatures shared by microbes, the PAMPs. Mannose receptors and scavenger receptors bind carbohydrates and lipids on bacterial walls. Phagocytosis follows, pulling microbes into vesicles that fuse with lysosomes, creating the acidic, enzyme-rich phagolysosome, where invaders are dismantled. At the same time, toll-like receptors act as alarms. TLR4 recognizes lipopolysaccharide from gram-negative bacteria. Other TLRs detect gram-positive components, double-stranded RNA, single-stranded RNA, or unmethylated CpG DNA. Each TLR has a leucine-rich repeat region for binding and a TIR domain for signaling. Engagement of TLRs activate the IKK complex, which frees NF-κB to move into the nucleus. From there, NF-κB induces inflammatory cytokines like TNF, IL-1, and IL-6. These signals spread outwards, increasing vascular permeability, drawing neutrophils into tissue, and amplifying defense. The system is strong but regulated. Without NEMO, NF-κB cannot activate, leading to recurrent infections. With overactivation, chronic inflammation and cancer can emerge. For viruses, another system is essential: the interferon response. Cytosolic sensors like RAGI detect unusual viral RNA and signal through MAVs, activating IRF3 and IRF7. This leads to secretion of type 1 interferons, IFN alpha and IFN beta. These cytokines bind receptors on neighboring cells, turning on hundreds of interferon-stimulated genes that block viral replication and enhance antigen representation. Specialized plasmacytoid dendritic cells act as professional interferon factories, flooding the system with antiviral signals. When hidden danger persists inside cells, the inflammasome is engaged. NLRP3 assembles into a large complex, activating caspase-1, which cleaves pro-IL-1 beta into active IL-1 beta. This cytokine exits through gasdermin pores, often leading to pyroptosis, a fiery inflammatory cell death. Unlike apoptosis, pyroptosis is loud and messy, designed to alert and recruit. When this system misfires, autoinflammatory diseases appear out of nowhere, from NOMID caused by NLRP3 mutations to gout flares driven by urate crystals. Finally, the foot soldiers of innate defense arrive, neutrophils. Guided by chemokines like CXCL8, they roll along vessel walls, adhere tightly, and squeeze into tissues. Inside, they deploy granules, unleash a respiratory burst, and sometimes cast sticky nets to immobilize pathogens. They are potent but short-lived, clearing infection quickly and then dying so that inflammation can resolve. Through all of this, the innate system shows its dual nature: fast, broad, and powerful, yet dependent on balance and resolution. Inflammation is necessary, but it must fade. Alarms must sound, but they must also fall silent. As you breathe deeply, imagine this as a symphony. Recognition is the opening note. Signaling cascades rise like melodies. Cytokines spread as harmonies. Neutrophils and interferons add urgency, while the inflammasome strikes its sharp chords. And then, as the infection is controlled, the music softens, fades, and returns to silence. This is the innate immune system, ancient, tireless, and always rehearsing, so that when the time comes, the performance is seamless. A system you carry with you night and day, keeping you safe, even now, as you drift into sleep. Now for the final part, we are going to do one final review of everything that we discussed today. Just a simple condensed review of me retelling this story in the most condensed manner. If you still haven't fallen asleep, don't worry about remembering any detail. There is no test and no exam. You just need to relax and let the information flow while you drift into the dream world. The innate immune system is the body's first line of defense once barriers are breached. Its strength is speed, broad recognition, and the ability to shape adaptive immunity. Recognition begins with pattern recognition receptors, or PRRs, that detect PAMPs, microbial signatures like lipopolysaccharide, mannose-rich sugars, or viral RNA. Macrophages use receptors such as mannose receptor and scavenger receptors to phagocytose microbes. Inside, phagosomes fuse with lysosomes, forming the phagolysosome, where acidity, enzymes, and radicals destroy the evader. Toll-like receptors act as signaling receptors. TLR4 recognizes LPS and signals through adapters that activate the IKK complex, freeing NF-κB to enter the nucleus. NF-κB induces cytokines like TNF, IL-1, and IL-6, which then cause inflammation and recruit neutrophils. Variability in TLRs affect human responses. Without NEMO, NF-κB can't activate, leading to severe infections. For viruses, the system relies on interferons. RIG1 detects viral RNA and signals through MAVs to activate IRF3 and IRF7. This induces type 1 interferons, IFN alpha and IFN beta, which make neighboring cells resistant to viral replication and enhance antigen presentation. Specialized plasmacytoid dendritic cells are professional interferon producers. Type 2 interferon, IFN gamma, is made by T cells and NK cells to activate macrophages. When intracellular danger persists, the inflammasome comes into play. NLRP3 assembles, activates caspase 1, and cleaves pro-IL-1 beta into IL-1 beta. Gasdermin D forms pores, releasing IL-1 beta and inducing pyroptosis, an inflammatory cell death. Overactivation leads to autoinflammatory diseases like NOMID or gout. The major effectors of inflammation are neutrophils. Chemokines such as CXCL8 guide them from blood into tissue. They use secretory granules, respiratory bursts via NADPH oxidase, and sometimes NETs, neutrophil extracellular traps, to immobilize pathogens. Neutrophils act quickly and then die, ensuring inflammation resolves. The 4 classic signs of inflammation are redness, heat, swelling, and pain. Controlled inflammation clears infection and heals tissues. Unchecked inflammation damages the host. In summary, PRRs detect PAMPs, which lead to phagocytosis. TLRs signal NF-κB, which then leads to cytokines. RIG1 and interferons block viral spread. Inflammasomes activate IL-1 beta and pyroptosis. And finally, neutrophils rapidly kill microbes and then resolve. The innate immune system is fast, broad, and essential, a bridge to the adaptive response, tuned for balance between protection and restraint. Now as you rest, breathe in and breathe out slowly, remembering whatever you can, or even refusing to learn anything. There is no correct answer. You are to relax and drift into the dream world. Thank you for listening. Good night.